Hi, I'm Levi Waldron, and I'm just going to briefly introduce our resource BugSigDB and its application to Parkinson's disease. BugSigDB is a Semantic Mediawiki, the same technology that powers Wikipedia for recording information from the published literature. It focuses on signatures of differential abundance from microbiome studies. So this main page here gives a bit of an introduction to the project and some of the different major things that you can do with it. There's also a search and a help page, which is actually a lot of good information here in this help page for contributors, users, and administrators and reviewers. Some of the things, these are just examples of data stored here, the featured taxon of the day, 2 featured studies of the day, some statistics to give you an idea of how large the database is. As of March 6th, 2025, there have been 178 contributors to the database. These are editors and reviewers, and 1,300 studies, 7,000 signatures, 44 host species. We can use the taxa to look for some particular taxon of interest and see everywhere that it is reported. And this advanced search I'm going to demonstrate right now to find what we have here in the way of Parkinson's disease studies. So I'm going to go here and search for Parkinson. And I'll just choose plain Parkinson's disease. These are taken from the experimental factor ontology. So there are some different variants of Parkinson's disease available here. I'm just going to do the simple ones. I could search for multiple ones of those. I'm just going to search for Parkinson's disease right now. And as you can see, there are quite a few. We have, I think there are more than 70 in total in the database. I could go to one of these as an example. The other thing I can do here is download CSV files of these. So if I download studies, I would get this full list along with additional information like their PubMed IDs, publication years, and titles. If I go to one of these, I think I need to hide this first. We will see an example of what is stored here. That's information about the study. We have what are called experiments, which has information about the experiment that was performed, such as the location, host species, body site, condition, the groups that were contrasted, sample size, sizes, antibiotics, exclusion criteria that were applied, the type of laboratory analysis, statistical analysis, results from alpha diversity analysis, and finally the signatures that were reported of differential abundance. And again, you could go back to here and download those results in a CSV format for all of them. There's also an R bioconductor package called BugSigDB for importing those into R, but you can also just use these in a spreadsheet. So as of March 2025, this is a subset of all of those studies to just the ones that are case control studies. One of the things that this makes straightforward to do is to compare across all of these signatures to count how many times we see a taxon such as Lachnosporiaceae across these studies. And we see actually fairly remarkable agreement, replicability of these results across many previous studies of Parkinson's disease. So if you are interested in using this, you can just go ahead and use it. You don't need an account. If you're interested in contributing something, that would be fantastic. You can add results from a study in progress or published and immediately have it appear with these and with the exports and make it possible to analyze alongside all of the other studies. And if you want to do that, just take a look in the help page. There are some instructions for requesting an account. You can request an account and get started, and feel free to let me know if you have requests for areas of expansion or anything else about BugSigDB. Thank you.